Hello everyone. Today we'll discuss the topic from the next unit, that is unit 3, 3.1, respiratory pigments, types, distribution and properties. Among the respiratory pigment, the hemoglobin is the most widely distributed among the animals. So first of all, we'll discuss the structure and functions of hemoglobin. Hemoglobin is a protein, as you know, and constituting one third of the RBCs. The 65% at the erythroblast stage and 35% at the reticulocyte stage, that is a developing stage of the RBC. The normal concentration of hemoglobin in the blood, that is in adult males, that is 13.5 to 16.5 gram per 100 ml or deciliter, and in adult females it is 12.5 to 15 gram per 100 ml or deciliter. Approximately 6.25 grams, that is 90 milligrams per kg of hemoglobin are produced and destroyed in the body per day. In hemoglobin, as the name suggests, there are the two components. One is a heme and the another one is a globin. The biomedical importance of hemoglobin is these heme proteins are characteristics of the aerobic life. Hemoglobin is important in binding of oxygen and its transport and delivery to tissues, which is required for metabolism. Then the next is a 2,3-biphosphoglycerate, which is produced in RBCs by a rapport lumbering shunt is necessary for the stabilization of hemoglobin conformation at quaternary level by holding the salt bridges, and is important from understanding of high altitude sickness and the changes that takes place in adaptation at high altitudes. In cyanide poisoning and carbon monoxide poisoning, these both are fatal because they combine and inhibit the heme protein cytochrome oxidase in electron transport chain and stop the cellular respiration. The conversion of hemoglobin to methemoglobin by sodium thiosulfate forms the basis of treatment of cyanide poisoning, as cyanide combines radially with the methemoglobin to form the syn-methemoglobin, which is not toxic and spares the cytochrome oxidase. The main function of RBC is the transport of oxygen from lungs to tissue, and second one, that is the transport of CO2 from tissue to lung. And for this function, or to accomplish this function, RBC has the hemoglobin. The synthesis of hemoglobin, it starts early in the proerythroblast stage and at 35% at the reticulocyte stage. The hemoglobin is found in RBCs. Its main function is to transport the oxygen to the tissues. When fully saturated, each gram of hemoglobin binds 1.34 ml of oxygen. In the structure of hemoglobin, if you observe, the two components are there. One is the globin having the 4 chains, and the heme molecule, it having the porphyrin rings with central metal iron, that is a Fe++, and the iron is the site of attachment with the oxygen. There are 4 heme groups, each attached to an globin chain. So, one hemoglobin molecule can carry up to 4 oxygen molecules. According to sequence of amino acid in the primary structure of each chain, there are 4 types of chain. One is alpha, then beta, gamma, and delta. In heme molecule, there are 4 pyrrole structures or pyrrole rings. The 4 pyrroles are linked with one another by methane bridges to form porphyrin, and this hemoglobin having the porphyrin rings. The synthesis of hemoglobin, where occurs the heme and globin, these two these are synthesized at two different sites in the cells. Heme is synthesized where in mitochondria, while globin is a protein that is on the polyribosomes. Then the structure of this heme, it having the porphyrin rings, then centrally placed the metal ion, that is a iron, and that is to which when the globin is attached it becomes a hemoglobin. That we'll discuss in the next slide. This is a structure of the hemoglobin, and the where the what these porphyrin rings or these are the pyrrole rings are attached, or the 4 pyrrole rings to which the iron or the metal iron is there, and here this is the globin. How the structure? This is the where the CH3 is given, or the where the M is written. Here this is the methyl group, and this P is written at that position. There is a propenyl group, and on where the V is written at the outside at that position, this uh, this one is a 1V, and this is a vinyl group. Means in the structure of hemoglobin, the three groups are there attached externally to these rings. That is methyl group, propenyl group, and the vinyl. This is a structure of the pyrrole ring, and it is a Fe or iron porphyrin compound. The porphyrins are complex compounds with tetrapyrrole structure. This is a porphyrin ring having the one pyrrole, then second, third, and fourth. These four rings or these 4 rings, it forms a pyrrole ring structure, and this forms a polypyrene rings. Then this is a exact structure of hemoglobin or how it looks. These are the pyrrole rings, that is a one, then second, then third, and the fourth pyrrole rings. All these pyrrole rings are forming the porphyrin rings, and there is a centrally placed iron or iron compound to which the water molecule is attached. These pyrrole rings are designated as 1, then 2, then 3, and 4, and this forms the porphyrin rings, and these rings are combined through the methane bridges and to form a polyporphyrin nucleus. Then if you observe the carbon atoms or the external carbon to this, you can observe this is a C1, then this is a carbon 2, this is a carbon 3, carbon 4, then carbon 5, carbon 6, then carbon 7, and carbon 8. Means to each what? To each pyrrole ring, the externally attached carbons are the 2. in first 1 and 2, in second 3 and 4, in third that is a 5 and 6, and this in last that is a 7 and 8. These are not involved in the formation of these methyl bridges. These four pyrrole rings. These are attached by these methyl bridges. That is a what? This is a methyl bridge, and is the alpha. Then this is a beta, and then gamma, and this one is a delta. Then these externally, which are not involved in any ring, externally that this one, this is the having attached to the what? This methyl group. One, then to this 3, that is a methyl group is there, then C5 that is to this methyl group is present, and this C8 means 1, then 3, 1, then 3, then then to 5 and 8. The methyl group is attached. Then regarding this vinyl group. Vinyl group, you can observe vinyl group at the what? This C2 position, and again this vinyl group you can observe at the C4 position. Means at the C2 and the C4 position the vinyl group is attached, and the remaining two positions, that is a propionic acid, where it is attached at the C6 and C7 group. C7 carbon atom, there is a attachment of the propionic group, and this at C6 and the C7 to this propionic group there is a attached the globin molecule. Also, the two amino acid, that is lysine and arginine. Lysine is attached to the C7 and the arginine is attached to the C6 position where. And this arginine and lysine, these are the two amino acids. These are attached to the globin molecule, and this globin is attached to this iron. This is a entire structure, or the how is the structure. These are the four pyrrole rings, and these four pyrrole rings, these are attached by the methyl bridges. And what are these? That is the first and second attached by the alpha bridge, then the second and the third attached by the beta bridge, beta methyl, and what this, that is a gamma, and this one is a delta. The outer carbons of four pyrrole rings which are not linked with the methylene bridges are numbered as 1 to 8, as we have earlier discussed. The methylidine bridges are referred to as alpha, beta, gamma, and delta. In addition to these, the hydrogens at position 1 to 8 are substituted by different groups in different compounds. In protoporphyrin 9, which forms a parent compound of heme, the positions 1 to 8 in pyrroles are substituted by either methyl group, vinyl group, or, or the propionic acid. And the iron molecule or Fe molecule in the heme. This is internally placed. The iron can form the 6 bonds, 4 with the porphyrin nitrogens plus 2 additional bonds, one above and one below the planar porphyrin rings. The two hydrogen atoms in NH group of pyrrole rings, that is 2 and 4, are replaced by the ferrous iron, which occupy the center of the compound ring structure and establishes link with all the 4 nitrogens of all the pyrrole rings. Then the Fe or iron, besides its linkages to 4 nitrogens of the pyrrole rings, it is also linked internally, that is fifth linkage to the nitrogen of the imidazole ring of the histidine of the polypeptide chain. And The sixth valence is directed outwards from the molecule and is linked to a molecule of water in a deoxygenated hemoglobin, and when hemoglobin is oxygenated, water is replaced by the O2. Means this in this the iron molecule, this is the how is the what this hemoglobin bind to the what or the this attached to the water molecule, and when the oxygen is there or the in the oxygenated state, this hemoglobin is attached to the oxygen and the water is displaced. So that is in carbon it is attached to or it forms the linkages with all four nitrogens of the pyrrole ring, and what the fifth linkage to the imidazole ring of the histidine of the polypeptide chain, and the sixth valence is directed towards the water molecule, and when it gets bind with the oxygen, the water is displaced by the oxygen. The propionic acid groups of 6 and 7 position of heme of third and fourth pyrrole are also linked to the basic groups of amino acid, that is arginine and lysine of the polypeptide chain. Means the propionic acid, that is COH group of 6 and 7 position of heme of 3 and 4 pyrrole are also linked to basic groups, that is the arginine and lysine of the polypeptide chain that we have discussed. This is the structure of the hemoglobin molecule, and you can observe the different chains, that is the alpha chain and the beta chains, and the centrally placed what the iron molecule, and this is a molecule that is a protoporphyrin and the iron. These are these four chains, that is a 2 alpha and 2 beta, that is a alpha 1 alpha 2 beta 1 beta 2, and this is a in deoxygenated state, that is a what it binds to the what T2,3 diphosphoglycerate, and oxygenated state it having the heme molecule internally placed. What are the functions of hemoglobin? The oxygen delivery to the tissues means it transport the oxygen up to the tissues. Then reaction of hemoglobin and oxygen, it forms the oxyhemoglobin, and oxygenation is not the oxidation. One hemoglobin can bind to 4 oxygen molecules, and less than 0.01 second required for oxygenation. Beta chain move closer when oxygenated. When oxygenated, the 2,3-diphosphoglycerate is pushed out. The beta chains are pulled apart when O2 is unloaded, permitting the entry of 2,3-diphosphoglycerate, resulting in lower affinity of oxygen. When fully saturated, each gram of hemoglobin binds 1.34 ml of oxygen. The degree of saturation is related to the oxygen tension or partial oxygen pressure, which normally ranges from 100 mmHg in arterial blood to about 35 mmHg in veins. In relation with oxygen tension and hemoglobin oxygen saturation, it is described by the oxygen dissociation curve of hemoglobin that we will discuss in transport of respiratory gases. The characteristics of this curve are related in part to the properties of hemoglobin itself and in part to the environment within the erythrocyte, including pH, temperature, ionic strength, and concentration of the phosphorylated compounds, especially the 2,3-biphosphoglycerate. This is the state of hemoglobin in the oxygenated form and the deoxygenated form. You can clearly observe the what that is this hemoglobin or the four heme molecules and to each heme molecule the one oxygen. This is the one heme molecule, another, then third and the fourth. To each heme molecule there is a one oxygen is binded, and therefore one hemoglobin molecule it having ability to carry the four oxygen atoms. And when the in deoxyhemoglobin or the when the oxygen is dissociated, at that stage, there is a this it is replaced by the word the 2,3-diphosphoglycerate gets bind to this, and this is regarding the oxyhemoglobin and the deoxyhemoglobin. Then this is the structure of the myoglobin and the hemoglobin. These are somewhat similar structures, and the myoglobin it is formed of one heme molecule and attached to one globin chain, and it is found only in the red skeletal muscles and cardiac muscles. It gives their tissue their characteristic color. Myoglobin has much higher affinity for oxygen than hemoglobin, so it is unable to release it to tissues. In contrast, hemoglobin reacts with oxygen reversibly to give it to the tissues. The myoglobin concentration is increased in blood in a disease that is called as a myocardial infarction, and in case of muscle trauma and myopathies. Thank you.